Okay, so today I'm going to talk about the role of autophagy in silicosis. So to begin with, silicosis is a mineral dust-based lung disease, which is characterized by the excessive deposition of extracellular matrix proteins at the alveoli, and this is caused by a prolonged exposure to silica dust. In class, we learned that the typical pathogenesis of silicosis is that alveolar macrophages attempt to engulf the silica fibers, and these fibers lyse the macrophage, resulting in an inflammation response, which triggers fibrosis. However, increasing research has shown that autophagy is involved in this pathway. So the basics of autophagy is that essentially the cell forms this organelle called an autophagosome that eats itself. The autophagosome is typically involved in cell death, but it does play roles in other situations. So let's just go over the basics of how it is made. So some upstream signals signal for autophagy to occur, and these protein dynamics result in a bilayer membrane. The membrane elongates and eventually engulfs cytoplasmic material, such as organelles, resulting in a mature autophagosome. This autophagosome then fuses with a lysosome, which degrades everything inside. So this is typically how cell death occurs, but I'm not talking about that. I'm talking about the pathogenesis of silicosis. So Liu et al. Wanted to study how autophagy is involved in uh, silicosis and what are the molecular mechanisms involved. To test this, they cultured macrophages and exposed them to 50 micrograms of silica per square centimeter or nothing. They then took these macrophages and performed fluorescent microscopy on them to determine where proteins of interest are and their expression levels, and performed a Western blot analysis on them to determine the amount of proteins of interest that are in the cells. So they first discovered that autophagy is induced by silica exposure, and to prove this, first they did a Western blot analysis of P53 and MCPIP1. P53 and monocyte chemotactic protein-induced protein 1 are proteins that are known to interact with each other in a pathway to induce autophagy. So they quantified the amount of these proteins over a certain amount of time after exposure to silica and found that these proteins were becoming more expressed as time went on, suggesting that autophagy is occurring since the pathway is being activated. To definitively say that autophagy occurred, they stained for this protein called LC3B, and that is a protein that is present when autophagy has occurred. This is why it's called an autophagy marker. And they found that this autophagy marker in red was way more expressed in the macrophages exposed to silica compared to the macrophages without it. So this demonstrates that, hey, autophagy is induced by silica exposure. The next discovery is that autophagy activates macrophages to attract fibroblasts. I didn't include this Western blot since there'd be way too much to talk about, but they found that the macrophages exposed to silica overexpress many cytokines way more compared to the control macrophages. These cytokines may possibly interact with fibroblasts, the main cell type depositing the extracellular matrix proteins in silicosis. So they wanted to see how these cytokines interacted with the fibroblasts. To test this, they did a scratch assay, which is basically they cultured these red stained fibroblasts on a matrix, and then on that matrix they scratched it with a micropipette and then placed macrophages in the middle. They wanted to see if these fibroblasts would go to the middle, and to quantify that they measured the middle. The smaller the middle, the closer they are to the macrophages. And as you can see on the right hand with this quantification, they found that the fibroblasts moved way closer to the center when the macrophages were exposed to silica than compared to a control. So these macrophages are overactivated, secreting these chemoattractant cytokines. And the cool thing about these cytokines also is that not only are they chemoattractant, they overactivate the fibroblasts to secrete more collagen. Again, this was shown on another Western blot, not shown, but it's just so much to talk about. So many interesting things. But here's a little pathway to summarize everything that I just said. So the macrophage is exposed to silica. This activates the MCP-1 pathway along with P53. And these two proteins interact to regulate some transcription, which causes autophagy. Autophagy overactivates the macrophage with some pathways that are really complicated to overactivate the secretion of these cytokines. These cytokines would then attract these fibroblasts and then transform their morphology into a myofibroblast, a more activated version. And these are the cell types that exacerbate silicosis. The cool thing that they point out, though, through this diagram is that if you target the amounts of P53 and MCPIP1, this could be a nice diagnostic tool to detect silicosis early before it gets really bad. So just a cool clinical relevance that they discovered here. So I know, yeah, that's it for me. So I'm just gonna debrief with my dog Marshmallow, and thank you for listening. Have a good day.